Right. So my name is Kevin Lancon. I'm a postdoc in Philippe Seguela's lab here at the Montreal Neurological Institute. And we mostly do a lot of research on the brain and how that modulates pain, as opposed to what maybe you're more used to as people talking about what's happening in the peripheral nervous system. We really focus on what's happening in the central nervous system. And here we're going to talk, I'm going to talk really about what's happening between dopamine and how that modulates pain, and how you may also be experiencing these side effects, maybe yourselves. So one common jokes we have in the pain field, if you work in the brain, is that pain is all in the head, right? You guys may have felt this yourselves, right? Where a small injury, like a small paper cut, can be extremely painful, yet not really cause much impact on your life. Whereas you have very severe, life-threatening injuries that can actually be masked by shock. So, for example, here on the right is an Australian surfer whose foot was just eaten by a shark. But as you can see, he's having he's hanging in there, right? So this goes to show that at the end of the day, no matter what happens in the peripheral, if you're getting a lot of pain, if you're not getting any pain, what ultimately decides how you feel will be what happens in the brain, right? So then the next question is where in the brain is pain really represented, right? So it's very simple for other senses because they're very well studied, right? Somatosensation, for example, is very well shown to be represented by the somatosensory cortex. This is a part of your brain that is very well mapped out to different parts of your body, right? This was done by Penfield in the very building that I'm in right now, right? And as you can see, there's a very good mapping of the somatosensory cortex and how that really implies to how this part of the brain is responsible for the sensation of your neck or your shoulders or your legs, right? Similarly, movement is very well characterized, right? So similar to the somatosensory cortex, there's the motor cortex, which represents really the aspects of movement, right? It really is responsible for the ones sending their signals down to your muscles. That's really going to cause movement in your body, right? Similarly, vision, once again, very easily and very, well, not easily, it's quite complex, but it's very well understood in the fact that you have light that's shining onto your retina that is then causing a cascade, which will then activate your visual cortex in the back of your head in the occipital lobe right here, right? So all of these are, for example, very well studied. So when we talk to people about pain in the brain, everyone's always asking, where is the brain? Where is the pain cortex? Where in the brain is pain represented, right? And now I'm showing you this picture, and I don't want you to get overwhelmed, although that's the entire point, right? It's that in the brain, there is no pain cortex, right? It really is a matrix between a lot of different brain regions interacting with each other and modulating how we feel pain, right? So unfortunately, it's not as easy as going as this area of the brain is dysregulated, and if we fix that, it's really a cure-all, right? It really involves the interaction between different brain regions and how they communicate with each other to really modulate the pain that we feel. So very quickly, I'm going to give an introduction to how pain is transduced throughout the body and where it ends up in the brain. So after you get a heat, a noxious cold, a chemical, or even a tactile painful stimuli in your periphery, for example, in your hand, this will then be transduced along your nerves to right outside your spinal cord in the dorsal root ganglia, which will then connect to other neurons that go up your spinal cord and will then go to different areas of the brain, right? This is how you feel things, pain included. So once again, do not get overwhelmed by this. This is just to show that it is in fact a lot more complex than I'm letting on, and that even though I'm going to simplify this talk quite a lot, it really is more nuanced than I'm showing, and it really involves the interaction between different brain regions, right? And in terms of pain sensation, in the brain, it is commonly divided into two different types. There is the sensory discriminative here in the top left, and then there is the affective component. So in other words, there is the emotional component and then the sensory component attached to pain, right? So if you hurt your hand, for example, you will feel the pain. That is the sensory component. But there is also an emotional non-sensory element that is also encoded by your brain that is also very prevalent. So out of all these brain regions that I'm showing here on the right, the one that we are going to be looking at is the ACC, or the anterior cingulate cortex. Now, this is a brain region that's involved in a lot of tasks, not just pain, right? It has a lot of roles into attention and memory processes, which we'll get back to later on. It plays a large role in decision making, reward-based learning, and mood regulation. And lastly, the affective components of pain processing, or the emotional component of pain processing. Right. So why are we looking at this brain region? Well, the reason we're looking at this brain region is that this is one of the most constitutively active brain regions in chronic pain patients and is very consistently activated by pain. So on the left here is fMRI data. So what fMRI data is, it's a scan of the brain, a functional MRI, that will look at the activity of different brain areas, right? And it does this by looking at pretty much how much blood is flowing in different areas of the brain, right? If more blood is flowing in one area of your brain, that means that the activity in this area is increased and it requires more support from the body, right? More blood. So what we see here on the left is the ACC and the somatosensory cortex lit up or active when you give someone, a patient, a painful or a noxious stimuli, right? We can see that this brain region is quite active following this painful stimuli, right? And this has then been done in rodent models and in mice and rats, right? And we can actually even image the brain by putting a microscope in the brain of rats or mice that are freely moving. And we can image this area to show that when you give a pain sensation, you can see neurons are lighting up and really being activated by this pain, right? So the question is, what is happening in chronic pain, right? What is going wrong in this brain region that is most likely promoting the classical symptoms of chronic pain that you're familiar with? So to do this, first we need to talk about how we're going to look at chronic pain, right? Because although I do some human studies and some clinical trials later on, I am mostly going to be talking about what happens in rodents right now, because this is what we research, right? So first, how do we cause chronic pain in rodents? The simplest one is by causing neuropathic pain using the spared nerve injury. So one of the most common causes of pain is from a nerve injury, right? Through an accident or things like that, or even diabetes, for example. Diabetes is neuropathic, right? It will actually cause the decay of your peripheral nerves. Now, what we do in rodents is we look at the sciatic nerve and we just cut two branches of the, let me change my pointer here. We cut two branches here and here of the sciatic nerve to induce a hypersensitive area of the mouse foot, right? And this is a long-lasting condition that will last throughout the life of this animal from this point on, right? And it will really cause a very high increase in pain. So how do we measure pain in these mice, right? The simplest way is by doing something called a von Frey pain threshold test. Now, what a von Frey test does is it puts a mouse inside a cage with a grill bottom, and then we have a little pokey stick that comes in that will poke the paws of the mice. Now, if a mouse is more sensitive to pain, you can only put so much pressure before the mouse removes its hand, right? But if the mouse is more resistant to pain, you can put more pressure and then the mouse will move its hand, right? So on the right here is just the withdrawal threshold. And I'm going to show another one of these later on. This is why I'm explaining this. So the lower your withdrawal threshold is, the more sensitive you are to tactile pain, like this test, right? So for example, sham, which is our control group, has normal tactile thresholds, but in the neuropathic pain group, you see that this actually goes down after the procedure that I talked about previously. So going back to the ACC, why are we looking at this brain region and what is going wrong in chronic pain? So starting on the left first, this is once again fMRI data. And what we see is that following chronic pain onset, or in chronic pain patients compared to control patients that do not have chronic pain, the activation of this prefrontal cortical region, the ACC, is dramatically increased in chronic pain. As you can see, following a painful stimuli in these patients, you can see that the activation of the ACC is much higher in chronic pain patients compared to other patients, right? And when we want to translate this into animal models, like I talked about previously, we can, right, doing the sham or the SNI procedures. Now, I don't want you to get confused by this data. I'll explain what this is in a second. But on the left here, right, this is just the overall activity of this brain region based on blood flow. And on the right here, this is the actual activity of the neurons themselves, right? So just looking at the density of the bars at the top, right, you can see that in the SAM conditions, there's less bars than on the SNI conditions, right? So we see that both the activity of the entire region is increased, but the activity of the neurons themselves are also increased in neuropathic conditions. Now, why am I talking about the ACC, right? Well, the simplest solution is that this brain region is dysregulated by chronic pain. And what's very interesting is that if you inhibit this region that is hyperactive, you can actually stop a lot of the symptoms of chronic pain, right? So here, once again, this is a von Frey test that I'm showing, just like I talked about before. The red animal is the one that is in a neuropathic condition or a chronic pain condition. And then the red line is a control animal. So what we see, and this has been done through other experiments, not just this one, is that whenever you see the yellow bar is when they inhibit this region through a special mediator, right? But they inhibit the general activity of this region. And we can see that the pain thresholds actually increase when you inhibit it, goes back down, increases again, and goes back down, right? So you can very easily modulate the symptoms of chronic pain simply by changing the excitability of this one brain region. Right? So this region has a lot of powerful control over how you feel pain, both from a sensory perspective and an emotional component, right? So the next question is, why is this brain region hyperactive, right? Why do we see this increase in chronic pain conditions? What is causing this? And this is where we're finally going to get into the dopamine part of the talk. So a lot of you are most likely familiar with what dopamine is. It is the reward. It is the reward signal, right? It is the reward chemical that your brain releases whenever you do something pleasurable, or you get a piece of candy, or something that is like gambling, for example, right? But it does a lot more than that, right? And some of you may be familiar with its effect in Parkinson's, right? Where it has a lot of effect on fine motor control. But generally, the way the dopamine system works in the brain is that you have this nucleus right here in the midbrain called the VTA, or the ventral tegmental area. And it will send these projections, or these cells will then release dopamine into different regions of the brain, right? Now, the ACC is right here. And we have a lot of dopamine that's coming in from the midbrain to this brain region, right? So we know that there's a lot of dopamine in this brain region, and it's most likely having an effect on the brain, right? So what's very interesting about the ties between dopamine and pain is that there is actually a lot of comorbidities between low dopamine in the central nervous system and high pain sensitivity, right? So for example, Parkinson's patients have an extremely high occurrence of chronic pain, right? And if you give these Parkinson's patients, so for those of you that are not familiar, in Parkinson's, one of the main causes of Parkinson's symptoms is the fact that dopamine cells are in fact dying in the brain, right? They are toxic and they are just no longer functional. So this death of these dopamine cells results in a lower level of dopamine in the brain, right? So these patients have low dopamine and they have a lot of chronic pain, right? So on the right here, this is once again fMRI data. And this signal right here is the ACC once again. Now, what's interesting is that if you give a PD as a Parkinson's patients and they are off their medication, they have a high increase in chronic pain and they have a high level of activity in the ACC. Now, when you give medication to a Parkinsonian patient that has chronic pain, PD on, and this activity decreases and the symptoms of chronic pain go down as well, right? Now, the medication that we typically give to Parkinsonian patients is something called L-DOPA or other medications that resemble that medication. And what L-DOPA essentially is it's a precursor to dopamine. So if I were going to give you L-DOPA, your body would simply be able to produce more dopamine and you would have more dopamine, right? So in Parkinson's patients where they have less dopamine in the brain, you give them this medication and this will increase the amount of dopamine that they are getting. Now, this goes slightly beyond Parkinson's, though. In fact, there is a very high correlation between this, in fact, right? Like I said, when you have low dopamine in the central nervous system, you have a lot of chronic pain. But when you have high dopamine in the nervous system, you actually have a very, very strong insensitivity to pain. So, for example, in schizophrenia, schizophrenic patients generally have a high level of dopamine in the prefrontal cortex, and they have pain insensitivity in a lot of cases where they are just not aware of the pain they are feeling, right? Now, this can also extend beyond just mental diseases, right? So for example, cocaine or amphetamines are drugs that strongly increase the level of dopamine that your body can feel, right? And those are well known for being quite strong analgesics. So there seems to be a very large trend between dopamine and pain, where less dopamine is more pain and more dopamine is less pain, right? So given the fact that the dopamine seems to have such a strong role in modulating the pain of the body, we wanted to know how it affects the ACC, or this brain region that has such a strong, powerful hold over your pain sensitivity as well. And like I mentioned before, right, in the brain, you have a very strong pathway that releases dopamine into this region that we're looking at, right? So what is the role of dopamine in regulating this? So to do this, we have to do something called electrophysiology. Now, I showed these results earlier when I showed you the spikes. What this essentially is we need to be able to measure brain activity. Now, it's in patients, you can do this through fMRI, but fMRI, once again, just looks at the overall activity of this brain region when it comes to blood flow. But we need to determine the activity specifically of the neurons themselves. Now, to do that, what we do is we get a mouse. We will get the brain out of the mouse and we'll slice it up into very thin slices. Then we will get the neurons and we will put an electrode into the neuron that will let us measure the electrical activity specifically of this neuron, right? So for example, here you can see that you have very high activity in this neuron. Once you are inside of it, you can record the electrical activity, right? So for example, here what we did, and don't get bogged down in the details, right? This is very general. But the general gist of it is that when we add dopamine to these neurons, the density of the spikes go down, right? So when we add 10 micromolar dopamine, which is a lower concentration, we get a slight decrease. But when we add 5 times this concentration, we get a very nice inhibition, right? So we can see that we have a dose-dependent role of dopamine inhibiting these neurons, right? So we know that dopamine is going to this brain region. We know that dopamine inhibits this brain region, and we know that this brain region is hyperactive in chronic pain. So the next question really is, what is happening to this dopamine signaling in chronic pain, right? Are we just losing this dopamine? Or are we just having less dopamine going to this brain region? And that's really the whole work that we're trying to do here. So one thing we wanted to test is what happens if you just give more dopamine to the mice, right? So the way to do this is we use a test called the condition place preference. Now, like I mentioned earlier, one of the tests we do to measure pain sensitivity is just the von Frey assay, where we take a hand, we put a needle, and we see how much the mouse can support before moving its paw, right? Now, this is a very rudimentary test, right? You're just looking at mechanical threshold and test and pain sensitivity. And this does not really apply to patients such as yourselves, right? Because what's interesting is that in chronic pain patients, what seems to be most of the issue is the affective component of pain. And in fact, your traditional painkillers, such as gabapentin or even opioids, tend to not really even change this aspect of pain. You will still be as sensitive before and after. But what it does affect is how your body internalizes that pain. Now, we can't ask the mouse how they're feeling, so we have to use another assay to determine how this works. And we do this using the condition place preference. So it's very simple. We put the mice in a 2-chamber box, right? And we have the mouse learn that in one chamber, it has an analgesic. And in the other chamber, it has no analgesic. Now, then we can put the mouse back in the box and we can see which chamber it naturally goes towards, right? If it naturally goes towards the chamber with the analgesic, that seems to show that they prefer this chamber unconsciously because they most likely felt the analgesia and the relief of pain in this chamber, right? So, like I said, we want to determine what dopamine does to this brain region in vivo, or in mice that are freely moving, to determine if this is really an active modulator of pain, right? So this is, for example, this is just the test that I previously talked, the condition place preference. And in here, what we're doing is we're just measuring the amount of time that the mice spend in one chamber, right? So in the top here, what we do is we make it so that one chamber is paired with dopamine. So we inject dopamine in the cortex of these mice, in this chamber, and we see if this is rewarding to them, if this is analgesic, right? And we see that in mice that are in chronic pain, the SNI mice, they spend a lot more time in this chamber, indicating that it is quite strong analgesic. Now, we also wanted to double check if this will occlude or stop the effect of other analgesics. So I'm sure a lot of you are familiar with gabapentin. It's generally known as the first line of defense for chronic pain, and it works quite well. But what we wanted to determine is if we block dopamine in this brain region and we give the mice gabapentin, will they still go to the chamber that is paired with the gabapentin? And we find that if we block dopamine, we no longer get this place preference, right? So this implies two things. One is that dopamine in the ACC is analgesic. And two, you need dopamine in this brain region to have effective analgesia. So this is a critical and core pathway of the brain that the body uses to likely mitigate or modulate how you feel pain, right? So the next question really is determining how is chronic pain decreasing dopamine output, right? How is chronic pain going to be affecting this? Because we know that dopamine is inhibitory, and we know that this region is excitatory, is increased excitability in chronic pain, right? So we wanted to determine really how is dopamine changed in the brain in chronic pain, right? And so to do this, what we do is we take mice and we will attach an implant to them, right? So this is a mouse that is freely moving. And what it has is an implant in it inside the ACC, or the anterior cingulate cortex, that can measure really the dopamine in this brain region, right? Which is really great. Technology has gone really far in the past 10 years, and it allows us to do these really complex things where we can measure the biosensors, the dopamine in this brain region in mice that are freely moving, right? And so what we see, and I'll keep it very simple, right, is that if you condition the mice that are, once again, attached to this, that can measure the dopamine, and you condition them to get a sucrose reward, very simple, right? This is the best way to get dopamine in the brain, is to give something rewarding. We see that the mice that get the sucrose will have a very nice release of dopamine, right? But if you give these mice chronic pain, this is greatly decreased. And you can see this just by the amount of yellow in this picture, right? You can see that in control conditions, you have much more dopamine, but then in chronic pain conditions, this goes down. So if you just graph it, don't get too bogged down in the specifics here, but what this generally shows is that in chronic pain, you have less dopamine in this prefrontal region, right? And this is actually very well established now in both rodents like this and in human patients as well, right? They've done a lot of fMRI imaging that look at the midbrain and the dopamine centers, and they see that in chronic pain patients, this tends to be severely inhibited, right? And this could be one of the reasons why there's a lot of comorbidities between low dopamine and pain, right? Not just Parkinson's, but depression as well, right? There's a lot of different circumstances where decreasing the dopamine in your brain actually makes you more sensitive to pain. So if you remember earlier, I talked about the role of Parkinson's disease and how it has a lot of pain. But if you give these Parkinsonian patients L-DOPA, or more dopamine, it tends to decrease pain sensitivity. So what we wanted to determine is, can we give L-DOPA to mice, or even humans, really, to decrease pain, right? And so once again, what we did is exactly that, right? So don't get too bogged down in the specifics here, but what we're generally showing is that if you compare the blue line here to the red line is the chronic pain condition, and the blue line is the chronic pain condition plus L-DOPA. So you see that the activity of the neurons themselves are decreased. And if you look on the right here, this is the pain thresholds, we see something the same, right? So we see that in SNI, you have a much higher pain sensitivity. But if you give these SNI mice L-DOPA, the blue line here, you see that this actually alleviates a lot of the problems that you see with chronic pain. Now, something else that we saw that was very interesting is that, once again, like I talked about with gabapentin, if you block dopamine signaling, you block the effect of gabapentin. But if you also give L-DOPA, you also block the effect of gabapentin, right? So this may be confusing, but what this actually states, and it's very important, it's that L-DOPA is essentially doing the exact same thing as gabapentin, right? It is simply increasing the dopamine in your brain that is causing analgesia. right? Very simply. So we did this in mice, right? And we have there is a doctor in the States, right, that really liked this story, and he wanted to determine, can we use this in humans, right? So he actually did a clinical trial with L-DOPA, and because L-DOPA is now an established drug, right, you don't need to go through the phase 1, phase 2, phase 3 clinical trials. You can just give this to patients and see what happens, right? And what they saw is that the L-DOPA actually works moderately well for chronic lower back pain, right? So in the left figure here, the LDP plus NPX, what this is it is L-DOPA plus naproxen, or it's an NSAID. It's just an anti-inflammatory drug, essentially, like ibuprofen. And so it's just an anti-inflammatory. And what we see is that it works pretty much the exact same, exactly the same. If you give L-DOPA and naproxen, it works exactly the same way as if you just give a control plus naproxen. But in females, it works surprisingly well. So we found that in females, the effect of L-DOPA is stronger than the effect of naproxen, right? They found that it's actually a greater analgesic in females than in males, and it works surprisingly well, right? So this is, once again, a proof of concept clinical trial to show that this could be something to look at. Now, doctors are not going to prescribe you L-DOPA like that, because L-DOPA, although it's very good, it has its uses. It is very good to stop the progression of Parkinson's disease. But after a very long time of being on L-DOPA, what happens is your brain can no longer differentiate between low and high dopamine signals. So you actually get an extremely high side effect of this long-term L-DOPA is called dyskinesia, or very uncoordinated movement, similar to Parkinson's symptoms, right? So although this works quite well in a proof of trial concept, we are going to need a more specific approach to really limit the side effects, right? So in the final parts of this talk, I would like to talk about how this is happening, right? I've mentioned a lot about how the ACC is modulating pain and how dopamine is involved in modulating this region and therefore pain. But to people, this doesn't make a lot of sense, right? Because in the periphery, it makes a lot of sense, right? If I give you lidocaine, lidocaine is a nerve blocker. So if I give you a nerve blocker in the arm and I stimulate your hand, the signals will not be able to go down your arm. So you will not feel pain. That is very simple. But if I give something to your brain, how is that affecting pain sensation, right? Because your periphery is still sensing it. You are still getting these signals that are going to your brain. So the best way to explain this, I think, is to understand really what the value of pain is. So pain is a signal that you will feel, but what is this purpose? And people can argue this, right? This can be philosophical, but generally one of the best values of pain is that it's a form of attention stimulus. It'll focus your attention on the part of your body that is damaged or in danger in order for you to move that body part away from the danger, right? And so what it really is, the pain is really just an attention indicator. It is a way for your body to say, hey, there's a flame here, move your hand away. That's essentially the role of pain, right? So if I remove pain from someone, and this unfortunately happens, right? There are people that have what's called a congenital insensitivity to pain, where these are people who cannot feel pain because they have slight mutations in the way of the way signals are transduced. They just cannot feel pain. Now, unfortunately, these individuals generally die before they reach maturity because they have no signal in their body that tells them to watch out for pain. So if they have a broken bone, they don't feel it. They do not know that they have internal bleeding that should be looked at, right? So this just causes more bleeding, and eventually they will die of causes like this. If they put their hand on the radiator, they will not feel the burning. They will keep their hand there until it is burned, right? So although pain is a nuisance a lot of times, it is really a critical survival signal that we need, right? But the problem is that it will get dysregulated sometimes. For example, in the case of chronic pain, where the injury site is fully healed, you still feel pain. Why does this happen? Right. So this is what we're going to talk about here. But first, we really need to determine the value of pain and what its role is in making us feel pain and how it really functions for us. So earlier I mentioned that we're looking at the ACC, and it has a lot of roles. right? And up until now, we talked about the active components of pain processing. But what the ACC also does, and this is actually its main role, is its role in attention and memory processes, right? It is very important to tune our attention and to deflect our attention from task A to task B, right? So the way I'm going to show this, and this is not our results, right? These are results done by people in great labs. And what they show is that if you get a monkey and you put them in a chair and you put electrodes in the brain and you record the activity of neurons, and then you show them this. So this is essentially a white screen. Then they show a picture A, a white screen, a picture B, and then they will ask the monkey to hold this on a white screen. So the monkey has to remember what's going on, right? And then there is another screen that comes up that asks, which picture did you see first? And they have to say yes or no. So what this requires is that for 2.5 to 2.8 seconds, for about 3 seconds, these primates need to recall what they saw without having a picture in front of them, right? So this is called working memory, right? If I give you a sequence of 5 numbers and I showed them to you and then I took them away, your brain has to remember these 5 numbers very well without them being in front of them, without putting them into your long-term memory, right? It has to keep them in your mind. It's sort of like the RAM of your computer. It's the brain, it's the working memory, right? It's really the short-term memory that goes away after you don't need it anymore. So here they're looking at the activity of the ACC, and they can see that when they are requested to hold the image in their brain, you can see that the neurons increase in activity to support this effect, right? So the ACC plays a huge role in modulating our attention and keeping things in our mind, right? Keeping those phone numbers or those numbers in your mind without them being in front of you. So this is work that was done by Amy Arnsten. She is a world expert in this type of studies. And what they found is that the level of dopamine you have in your prefrontal cortex determines very heavily how you can focus on things, right? So if you have too little dopamine, what happens is your attention is generally sensitized. So everything picks up your attention and you can't really focus on anything, right? You always have distractors that are coming in, showing you something, and then you change your attention and so forth and so forth. If you have too high dopamine, you cannot maintain your attention on anything, right? Your attention is essentially inhibited. So something that's telling you to call your attention, you will not even pay attention to it. You won't focus on it, right? Until eventually you do. So there is what we call an inverse U-shaped curve, right? Where you want really the optimal level of dopamine in your brain to have a good attention. Too low is bad and too high is bad as well. So how this translates to pain is that chronic pain patients, such as yourself, will very commonly report problems in attention and working memory, right? The ambulance that's passing by will be extremely aggravating, right? It will stop you from concentrating. It'll keep your mind distracted. You won't be able to focus. And this is just a side effect of your attention essentially being sensitized, right? So how this translates to pain signals is that in a healthy subject or a control subject that has no chronic pain, they will feel pain, and then it'll transduce and it'll go to their brain and they will think about it. Oh, that's hurts, and then they'll forget about it, right? But someone with chronic pain where they have decreased dopamine, they will not be able to forget about it. That pain will stick on their mind. It will constantly come at them. They'll always be paying attention to these pain signals, right? So this is actually one of the bases for ADHD, where ADHD patients will actually have too little dopamine, so they can't focus on anything because they're constantly being distracted. And in fact, a lot of ADHD patients have a lot of chronic pain because of this, because they are constantly focusing their attention on the pain signals, right? They're not effectively filtering that information out. So while I close up here, I just want to talk about one more thing, and it's that throughout this entire presentation, I talked about dopamine coming from this nucleus here that goes towards the prefrontal cortex. But this is, like I said, much more nuanced than just dopamine, right? You have also serotonin, which is 5-HT. You have norepinephrine, which is here as an NA, and acetylcholine, right? All 3 of these regions send inputs towards this brain region, the ACC. And what this will do is they are very involved in modulating how this brain region focuses our attention, right? So, for example, looking at norepinephrine has the exact same curve as dopamine, right? You need an optimal level of norepinephrine in your brain to have functional attention. If you have too much, that's bad. If you have too little, that's also bad, right? So you really need to make sure that you keep all of these neuromodulators in line. So any illness or any condition that affects the release of norepinephrine or dopamine will affect your attention, right? Depression, for example, with low dopamine tend to have problems in attention and they have chronic pain. ADHD, they have problems with attention and problems with chronic pain. PTSD, likewise, they have problems with attention and problems with chronic pain. Problems with attention generally go towards problems with chronic pain, which is very interesting in and of itself, right? So the last slide here, what we want to show is that instead of thinking just dopamine, what's really happening is that it is a concerted effort between these three monoamines, 5-HT, norepinephrine, and dopamine, to really tune the network of the ACC and give you proper sensory discrimination, right? So the three neurons that are firing will be able to differentiate between these three neurons. In chronic pain, what's happening is that we're really dysregulating the level of dopamine, which is decreased, the level of norepinephrine, and serotonin, to make so that now all signals that you are processing are viewed as painful. Your body can no longer effectively filter out the painful from the non-painful, and everything is just painful, right? So we're doing a lot more work in this area to determine really what's going on, but the general gist of the matter is that what's happening in the central nervous system is much more complicated than what happens in the periphery, because it involves all these brain regions that not only work on pain, they work on other areas, right? And so we are trying to really look at the specifics to determine what's going on in the brain of these patients, right? To really try and find better ways to treat chronic pain, right? And that is the end of the talk. I'd like to thank my lab, obviously, and our collaborators. Of course, the QPRN, who has helped tremendously, and the Alan Edwards Institute for Pain, right? And the neuro, obviously, McGill, and all these funding sources that have been great throughout our research. And with that, I will take any questions you might have.